Hi, I'll be your doctor today. So, what symptoms have you been having recently?
I just have really bad chest pain. I've been coughing up blood, and phlegm, as well, like very bad shortness of breath, and like just overall tightness in the chest. But I was thinking, my grandma was diagnosed with cystic fibrosis when she was like a couple years old. Is that a possibility?
Yeah, it is. Cystic fibrosis is a genetic disorder that can be passed down generations. We could run some tests to see if you have cystic fibrosis.
Sounds great, doctor.
A few moments later.
Hi, so I got your test back, and it seems like you do have cystic fibrosis. You have the mutation that gives you cystic fibrosis. So some treatment plans that we can start are some gene therapy, or we can use CRISPR to cut out and fix the mutation within your cells and see if that helps alleviate any of your symptoms.
Sounds like a great plan, doctor. Well, doctor, I just feel so much better. Like, I haven't been coughing up blood. I haven't had any symptoms. I can just breathe so fantastically.
Nice, that's really good. That means that the CRISPR has successfully fixed your mutation, and now your cells are being able to work properly, and now you won't have cystic fibrosis anymore.
Yay!
Yay!
And now for our presentation on CRISPR and its uses in medicine.
What is CRISPR/Cas9? CRISPR is short for clustered regularly interspace palindromic repeats. Now, this gene editing technology has allowed for errors to be corrected in the genome and allowed for us to turn off and on genes. Now, how does this process work? This process works by first identifying a sequence of the genome that has an issue and problem and creating a specific guide RNA to that sequence. Now, this guide RNA is attached to a Cas9, which cuts the specific region that guide RNA led it to. This is the problematic region that is cut it out. Now, the complex of the guide RNA and Cas9 is introduced to target cell, and it locates these specific problematic regions and cuts them out of the genome. Scientists can then edit the genome by modifying via insertions of new sequences or deletions of these problematic sequences. Now, CRISPR-Cas9 is used within many contexts of the real world. For example, we can use it to correct genetic disorders that are caused by single gene modifications, for example, cystic fibrosis. We also see its use in the treatment of HIV, and it's able to do this through targeted HIV-1 genome activity. Now, we also see its use in engineering somatic cells to create cancer treatments and develop these treatment methods. Now, there are a lot of limitations that come with CRISPR. These include the delivery of gene editing to the right cells. Also, off-target effects, which can cause unintentional edits to other parts of the genome that are not supposed to be edited. And also, there's controversy in genome embryo gene editing because, of course, we're editing embryos. And that can cause religious controversies as well as people not being in agreement with the edit.
Let's talk about the history of CRISPR-Cas9. In 1987, researchers at Osaka University found a series of repeating DNA sequences in E. coli. However, they did not know the purpose or function of these repeats. In the 1990s, similar sequences were found in other bacteria and archaea. Scientists started to notice a pattern with these repeats. There would be a repeat and then a unique spacer of DNA sequence. In 2002, the name clustered regularly interspaced short palindromic repeats, or CRISPR, was coined. Scientists also identified an associated gene nearby called Cas. In 2005, researchers discovered that the spacers within CRISPR matched the DNA of viruses that had previously infected the bacteria. In 2007, Felipe Horath showed that these matches in spacers was not a coincidence. In fact, CRISPR can be used to protect bacteria from viruses. In 2012, Emmanuel Carpenter and Jennifer Doudna reprogrammed CRISPR-Cas9 to cut any DNA at any site using a custom guide RNA. In 2013, teams led by Feng Zhang and George Church independently showed that CRISPR-Cas9 can edit genes in human cells. In 2020, Carpenter and Dona were awarded the Nobel Prize in Chemistry for developing CRISPR-Cas9. In the present, the uses for CRISPR-Cas9 has exploded, but some notable ones would be in agriculture, where it's used to change the genes of plants, in medicine for various treatments such as sickle cell, and diagnosis, where it can be used to identify if someone has a disease or not. How does CRISPR-Cas9 work? So first, you need to find a desired target DNA sequence. Then you need to design a guide RNA that matches that target DNA sequence. Once you have your guide RNA, you can construct a CRISPR-Cas9 system by attaching a Cas9 protein to a guide RNA. In order to deliver this CRISPR-Cas9 complex into the cell, there are various methods, but some popular ones would be electroporation, viral vectors, or lipid nanoparticles. Once Cas9 is inside the cell, guided by the guide RNA, Cas9 will scan the DNA for a PAM sequence. When the target has been found and Cas9 binds onto the PAM sequence, Cas9 will make a double-stranded break in the DNA. Cells will then repair the DNA break. If you're trying to make a knockout for a gene, non-homologous end joining will be done. However, if you're using CRISPR-Cas9 to make precise edits, such as fixing a mutation or inserting a gene, a template should be done and a homology-directed repair will be done instead.
Now, let's look at CRISPR applications for disease diagnostics. There are two main techniques that are used. The first one is with something called Sherlock, where it uses the Cas13 protein, which is an RNA targeting Cas protein, to look for specific RNA sequences that are associated with different pathogens. Essentially, you use Cas13 and then a guide RNA from one of those pathogens, and then a fluorescent RNA reporter, so that when the Cas13 finds that RNA, it releases the fluorescent reporter, telling us that that RNA is present in the sample. The applications are with virus detections, bacterial ID, DNA genotyping, and tumor mutation detection. There's also another version that has come out called Sherlock V2, which allows for multiplex detection, essentially allowing us to look for up to 4 viruses at a time. The other main type of technique that is used for CRISPR disease diagnostics is Cas12, which is a DNA-targeted Cas protein. Essentially, you use Cas12 plus DNA reporters, and it can detect things like HEV, which has been shown in clinical samples. The strengths of these kind of processes are that it's ultra-sensitive and specific, even to single base changes, it's rapid and low cost, and there's no special equipment needed. Another thing that's coming out soon is there are CRISPR kits in development for home testing. These will work for diseases like HIV and rabies. Next is CRISPR applications into gene therapy. Gene therapy is essentially when we edit genes in somatic cells to treat diseases caused by defected or mutated genes. This works for many different diseases because CRISPR allows us to go in and change and edit those sequences in order to correct the error that caused them. For example, monogenic diseases, which are diseases caused by a single mutation, are things like sickle cell disease, hemophilia B, cystic fibrosis, and retinal disorders. We can go in and correct the mutation that causes the disorder, thereby correcting the disease. It also works for some infection diseases like HIV because we can disrupt viral DNA, or we can knock out receptors which prevent the infection from being able to bind and grow. It also can work for cancer and can correct tumor mutations, inhibit growth, and boost apoptosis of cancer cells.
CRISPR has been used in cancer treatment as well. It has been used to edit immune cells in order to enhance immunotherapy. So in chimeric antigen receptor, or CAR, T-cell therapy, T-cells are engineered to recognize and kill cancer cells. And with CRISPR, the CAR gene can be inserted precisely into the T-cell genome, and it can also knock out genes that suppress the immune system or negatively affect the function of CAR T-cells. And this treatment has showed positive results in treating various types of cancer, such as leukemia, lymphoma, and multiple myeloma. CRISPR can also disable genes that help cancer cells avoid immune detection. It can knock out checkpoint inhibitors, which then unmasks the tumor and allows immune cells to attack it more effectively. CRISPR can directly target oncogenes and mutated tumor suppressors by editing out and repairing these genes in order to restore function. But this treatment is still in the early stages of development. And since CRISPR-based therapies aim for high specificity and precision, this reduces damage to non-cancerous tissues and healthy cells, which then reduces side effects.
And now for the ethical implications of CRISPR. CRISPR has received a lot of heated debate within the science community recently due to something called human germline editing. Human germline editing is where scientists can go in and edit the genomes of eggs, sperms, and embryos before they're even born. And obviously, because these changes are occurring in the DNA, these changes are actually inheritable. And so a very common case of this is in 2018, a Chinese scientist decided to take a set of twins and do germline mutations on them. Their names are Lulu and Nana. And he did this in hopes to make the twins HIV resistant by turning off their CCR5 gene, which is what HIV virus uses to enter the cells. Now, this sparked a lot of debate within the science community and for a multitude of reasons. The first one being, when you are turning off a gene, we don't always know exactly what's going to happen through this. Turning off one gene can lead to a failure of another, or can lead to some kind of other developmental issue. Secondly is, we understand that we can use CRISPR in a positive light and therapeutically to help treat diseases, but where do we draw the line in treatment? Where do we draw the line and say people can no longer use them for enhancements? And this can lead to a world where we have designer babies, where people can go in and decide, what they want their child to look like, their intelligence, appearance, and this can lead to a very dystopian-looking society. And with CRISPR being an expensive treatment, it can also be something that only is an option to the wealthy and not to the masses, even if it did become a more widespread option. This would lead to a greater divide, of course, in class and in health, but especially in a world where genes may come back and be more favorable again, this can lead to a greater divide in favorability between the wealth and those who are non-wealthy.
Here are our citations, and thank you for listening.